Right now, as you listen to this, you are legally, undeniably alive. I mean, you have a pulse, you're breathing, you're processing language.
Right.
But at this exact second, millions of cells inside your physical body are actively executing this highly orchestrated, programmed death sequence.
Millions of them, just right now.
Yeah, and then conversely, you could be lying in a hospital ICU, right? Yeah. Declared legally brain dead by every metric we have, yet millions of your constituent cells would be perfectly healthy. They'd be actively metabolizing, entirely capable of being transplanted into a stranger to live for, another 4 decades.
Which is wild.
It's totally wild. So what exactly is death? Like, are we a single living entity, or are we just this temporary container for trillions of smaller living entities that are constantly dying and regenerating?
I mean, that paradox is the absolute bleeding edge of modern biology right now. Because for centuries, we treated mortality as a philosophical absolute, like a cosmic decree.
Right, like a line you cross.
Exactly. You cross this distinct line from the realm of the living into the realm of the dead. But modern cellular biology has kind of completely obliterated that line.
Yeah.
When we stop looking up at the stars for meaning and we start looking through an electron microscope at our own tissues, the whole concept of a lifespan shifts. It goes from a mystical destiny to, well, a cold, hard problem of biochemical entropy.
A mechanical breakdown.
Right, a mechanical breakdown.
And mapping out that mechanical breakdown is our mission for today's deep dive.
Oof.
We're exploring the science behind a book that completely reframes how we think about our own physical expiration dates.
Yes.
We're doing an exhaustive analysis of a book called Why We Die: The New Science of Aging and the Quest for Immortality, written by Venki Ramakrishnan.
And Ramakrishnan's credentials make him, honestly, the perfect guide for this. He's a structural biologist who actually won the Nobel Prize for mapping the ribosome.
Which is no small feat.
No, not at all. The ribosome is that microscopic factory inside your cells that literally reads your genetic code and manufactures the proteins that keep you alive.
Yeah.
And because he spent his entire life looking at the fundamental atomic structure of life, he views aging through this deeply mechanistic lens. He doesn't see aging as this inevitable program written into our DNA to just, usher us off the stage.
Right, there's no grand biological clock ticking down.
Exactly. He sees it as the gradual mathematical unraveling of the incredibly sophisticated molecular machinery that allows a human being to function as a coherent whole.
So our goal here is to basically demystify that machinery. We are going to separate the hard-earned scientific realities of molecular biology from the very loud, incredibly well-funded hype coming out of, well, the Silicon Valley longevity industry.
There's a lot of hype.
So much hype. And we're going to explore what it actually means biologically to be what Ramakrishnan calls a temporary vessel. So whether you're listening to this to catch up on the latest biotech, or you just want to understand the engine under the hood of your own body, we're going to break down the sheer mechanics of why we age.
And I think the most compelling part of this exploration isn't merely the mechanics of how the body falls apart.
Right.
The true mystery, the evolutionary paradox that really drives the entire field of biogerontology is why we aren't programmed to live forever in the first place.
Because nature is capable of biological immortality.
And why?
In certain organisms.
Exactly. The real question is why humans were excluded from that blueprint.
Well, before we can zoom into the microscopic level to answer that, I think we have to establish why we are so singularly obsessed with curing aging in the first place. We have to look at the psychological reality of death.
Right.
Ramakrishnan argues that humans carry a biological burden completely unlike any other creature on Earth.
Yeah.
Which is that we possess the absolute conscious certainty of our own eventual demise.
Yeah, and we really have to differentiate this from the basic survival instincts we see in the animal kingdom.
Okay, unpack that a bit.
So if you watch a wildlife documentary, an antelope clearly exhibits fear when chased by a lion, right?
Sure.
But that is an acute flight or fight response to an immediate physical threat. Now, we also see complex emotional responses in highly social mammals, like elephants have been observed lingering over the bones of their dead, showing behaviors we definitely categorize as grief.
Right, there's a mourning process.
Yeah. But the human relationship with mortality is fundamentally different. It's a chronic, lifelong awareness. We don't just fear the lion in the grass. We fear the quiet, invisible degradation of time itself.
Which is terrifying.
It is. And Ramakrishnan attributes this unique burden to 3 specific evolutionary milestones.
Let's walk through those milestones because they really set the foundation for the multi-billion-dollar longevity industry we see today. The first milestone he identifies is the emergence of profound self-awareness.
Yeah. Self-awareness in early hominids was this cognitive leap that allowed an individual to recognize themselves as a distinct, singular entity existing on a linear timeline.
So you're not just reacting to stuff happening right now.
Exactly. You stop merely experiencing the immediate sensory inputs of your environment. You recognize that you have a past that you can remember and a future that you can anticipate. You become a subject in your own narrative.
Right. You realize I am a continuous thing. But simply knowing you exist in time doesn't automatically mean you know you're going to die.
Right.
That requires the second milestone, which is the cognitive capacity for generalization.
Right. Generalization is the engine of human logic. It's the ability to observe a pattern in the external world, abstract the rule behind that pattern, and then apply it to new situations.
Right.
So early humans observed the world around them. They saw the elderly weaken and succumb. They saw a hunter fall off a cliff and cease to function. And because they possessed self-awareness and because they could generalize, they made arguably the most terrifying logical deduction in the history of conscious thought. That cessation of life happens to them. I am fundamentally the same kind of creature as them. Therefore, that cessation of life will inevitably happen to me.
Wow. Yeah, that is a chilling realization to have on the prehistoric savannah. But it didn't just stay locked inside the mind of the one hominid who figured it out. And that's where the third milestone amplifies it, which is language.
Yes, language is the ultimate transmission vector. Once early humans developed complex language, this terrifying realization could be codified.
It could be talked about around the fire.
Exactly. It could be warned about, mythologized, and transmitted across generations. The fear of death stopped being an individual, private terror and became the foundational architecture of human culture, religion, and society.
And Ramakrishnan points out that this knowledge is so overwhelmingly heavy that it dictates our modern societal structure today. Like, we live in a state of systemic, willful denial.
Oh, absolutely.
We literally construct our physical spaces and our vocabulary to avoid looking directly at the biological reality of death. Just think about the euphemisms we rely on. We rarely say someone's biological function ceased. We say they passed away, or they departed, or we lost them.
And those linguistic choices are not accidental.
Right.
They subtly reframe death as a transition to another location or state of being rather than a final entropic collapse. It softens the psychological blow, and we see this exact same denial in our physical geography.
How so?
Well, for almost all of human history, death was a domestic event. People died in their homes, surrounded by multiple generations of their family. The physical reality of a decaying body was intimately known.
Yeah, you saw it happen.
But today, in the industrialized world, we have physically exiled the dying. We place them in hospitals, hospices, and specialized care facilities. We create this enormous physical and institutional buffer.
We curate our daily environment to maintain the illusion that death is an anomaly. Like, it's an event that happens to other people, in other buildings, under the supervision of professionals. It doesn't happen to us in our living rooms.
Precisely.
And this intense psychological tension, this desperate desire to look away, is really the primary driver of the longevity industry. We are terrified, so we want to hack the biology that limits us. But to hack death, we first have to rigorously define what it is. And historically, that definition was entirely binary.
Right. For centuries, the medical and legal definition of death was cardiopulmonary. It was a stopped heartbeat.
So no pulse, no breath, you're dead.
That was it. It was an indisputable, observable threshold. But human technological ingenuity intervened and kind of broke that definition.
With CPR.
Yeah. The development of modern cardiopulmonary resuscitation, or CPR, and the invention of defibrillators proved that cardiac arrest wasn't necessarily a final destination. It was a mechanical failure that could be temporarily reversed.
So the medical community had to move the goalposts. A stopped heart wasn't enough anymore, so they shifted the definition of death from the chest to the skull.
Yes. The clinical standard became brain death, the irreversible loss of all electrical and metabolic function in the brain and brainstem. But Ramakrishnan notes that relying on this modern standard exposes an incredibly peculiar biological reality about what a human being actually is.
Let's dig into that, specifically the biological paradox of organ transplants, because this is where the concept of a singular life completely unravels.
It really does.
Because a patient can be lying in an intensive care unit, hooked up to ventilators, they've suffered massive trauma and are legally, clinically declared brain dead. The person, the consciousness, the individual is gone.
Gone.
Yet the vast majority of the trillions of cells making up their heart, their liver, their kidneys, and their skin are actively alive.
They are completely viable. They're metabolizing glucose. They're synthesizing proteins. They're dividing. A surgical team can harvest a kidney from that legally dead donor, pack it in a specialized cold storage solution to slow down its metabolic rate, transport it across the country, and stitch it into the vascular system of a new host.
And it just works.
It does. That kidney will warm up, recognize the new blood flow, and continue to filter toxins perfectly for decades. The constituent cells did not die when the organism died.
And then you flip that paradox around to look at the living. As I mentioned at the very beginning, a living, healthy human is essentially a walking graveyard. You're experiencing millions of cellular deaths every single hour.
Yeah, and those deaths are not merely accidents or the result of environmental damage. The vast majority of them are meticulously programmed executions required for your survival.
Apoptosis, right?
Exactly, apoptosis. Unlike necrosis, where a cell is injured and violently bursts open, spilling toxic contents and causing inflammation.
Which sounds bad.
It's very bad. But apoptosis is a highly controlled dismantling. The cell shrinks. Its DNA is neatly chopped into fragments by specific enzymes called caspases, and the cell membrane forms little bubbles called blebs.
Blebs, great word.
It is. The cell essentially packages itself into neat little garbage bags, which are then quietly consumed by your immune system's macrophages.
And we rely on that programmed death just to take our basic shape. The classic example Ramakrishnan uses is embryonic development.
Right.
When a human fetus is developing, the hands initially look like solid, fleshy paddles. For you to have distinct, separated fingers, the cells comprising the webbing between those fingers have to execute that apoptosis sequence. They die precisely so the larger structure can thrive.
So we're forced to ask, if a legally dead body is teeming with healthy living cells, and a healthy living body is constantly executing millions of its own cells, what is the actual phenomenon of death?
Yeah, how do we define it?
Ramakrishnan offers a systems-level definition. He defines death not as the expiration of every individual cell, but as the irreversible loss of an organism's biological coherence.
Coherence, meaning the ability of the parts to communicate and work together.
Exactly.
Let's use an analogy to visualize this because it helps. Think of a legendary symphony orchestra. The orchestra is the organism. When the orchestra permanently disbands, that specific unified entity ceases to exist. The orchestra is dead.
Right.
But the individual musicians, the cells, they're still perfectly fine. They can pack up their violins and go play in another city symphony, which is essentially what an organ transplant is. The cell finds a new orchestra. The tragedy of death isn't the immediate loss of the parts. It's the permanent loss of the systemic coordination that allowed those parts to create complex music.
That's a perfect way to look at it. The magic is in the coordination. The signaling pathways, the hormonal feedback loops, the nervous system linking disparate organs into a single cooperative enterprise. When that communication network collapses irreversibly, the entity goes with it.
But if death is simply the loss of systemic coordination, why does the system lose coordination in the first place? Like, if the musicians are healthy and they know the sheet music, why does the orchestra inevitably break up? Is there a biological self-destruct timer embedded in our DNA?
You're asking if a genetic alarm clock rings at age 85, forcing the system to shut down to make room for the next generation.
Yeah, exactly.
Well, you are touching on the single most pervasive myth in popular understandings of biology, the idea of a death gene.
Okay, so it's a myth.
Complete myth. Ramakrishnan explicitly rejects this. Evolution does not select for a trait that actively kills a healthy organism just to benefit the abstract concept of the species. Natural selection operates primarily on the level of the individual passing on their specific genes.
So there is no programmed self-destruct sequence designed to clear the stage?
None at all.
So if there's no self-destruct program, why does the system inevitably fail? Why do we age?
To understand the failure, we have to look at how nature balances a metabolic checkbook. We have to explore the evolutionary economics of aging.
Economics of aging. I like that.
Yeah, Ramakrishnan draws on the foundational work of evolutionary biologist Thomas Kirkwood, who developed what is known as the disposable soma theory.
Let's break down the terminology. Soma is the Greek word for body. In biology, what exactly is the soma?
So your soma consists of all the structural and functional cells of your body. Your neurons, muscle fibers, bone cells, liver cells. It is everything except your germline cells.
Which are the sperm and the eggs.
Exactly. The germline cells contain the genetic information that gets passed to the next generation. Kirkwood's theory starts with the fundamental law of ecology. Resources are strictly finite. An organism operating in the wild only has access to a limited amount of energy, usually measured in calories extracted from food.
Every calorie is a metabolic dollar, and the organism has to decide how to spend it.
Precisely. The organism is forced into an evolutionary trade-off. It can invest its limited energy budget into rapid growth, reaching sexual maturity quickly and reproducing as much as possible. Or it can invest that energy into building incredibly robust cellular maintenance and repair systems for the soma, ensuring the physical body lasts for centuries.
But why not both? I mean, why not grow fast and build a maintenance system that lasts forever?
Because maintaining a complex biological system is unbelievably expensive in terms of cellular energy, or ATP.
Oh, okay.
Continuously proofreading DNA for errors, destroying misfolded proteins, replacing damaged tissues. It requires a massive, constant caloric burn. And we have to look at the environment where this budgeting evolved. The wild is exceptionally lethal.
Very true.
An organism faces constant threats from predators, starvation, freezing temperatures, infectious diseases.
So the external mortality rate is astronomical.
Think about a wild field mouse. The statistical probability of a field mouse surviving a predator strike, a harsh winter, or a bacterial infection for more than a year or two is practically zero. An owl or a snake will inevitably catch it.
RIP to the mouse.
Right. Therefore, from the cold, calculating perspective of evolution, investing massive amounts of precious energy into building a mouse soma with perfectly replicating DNA and flawless protein repair mechanisms that could last 200 years. Well, it's an evolutionary blunder.
It's a total waste of metabolic resources.
Exactly. The mouse is going to be eaten in year 2, regardless of how perfect its cellular repair enzymes are.
So the winning evolutionary strategy in a dangerous environment is basically to spend all your money early, build a body as fast as possible, make it capable of reproducing immediately, and don't bother paying for premium, long-lasting biological parts.
That is the essence of the disposable soma. Your physical body is a marvel of engineering, but biologically speaking, its primary function is to serve as a temporary vessel.
A fleshy taxicab.
I love that. Yes, a fleshy taxicab whose sole evolutionary purpose is to protect, transport, and successfully propagate the truly immortal biological material, the germline DNA. Once you have successfully reproduced and ensured the survival of your offspring, the evolutionary pressure to maintain the taxicab drops to near zero. You enter a state that biologists call evolutionary neglect.
Evolutionary neglect. So evolution essentially hands you the keys, says you've passed on the genes, my job here is done, and walks away. Your warranty has expired, and there's no evolutionary selective pressure keeping your cellular mechanics tuned up.
That lack of selective pressure is the passive cause of aging. But Ramakrishnan introduces another theory, proposed by evolutionary biologist George Williams, that shows how evolution actively works against us in our later years.
Uh-oh.
Yeah, it's called antagonistic pleiotropy.
Okay, that is a dense term. Let's pull it apart. So pleiotropy refers to a situation where one single gene controls or influences multiple seemingly unrelated traits in an organism. And antagonistic means those traits are in conflict with each other.
You got it. Williams hypothesized that there are specific genes that provide a massive survival and reproductive advantage when an organism is young. But those exact same genes, performing the exact same function, cause severe, detrimental, tissue-destroying effects when the organism gets old.
Let's use the concrete example from the text because it really helps.
Yeah.
Think about the biological process of calcium deposition.
Okay.
Imagine a specific gene that aggressively promotes the extraction of calcium from your diet and aggressively deposits it into your tissues. When you're a young hominid trying to survive in a harsh environment, this gene is incredibly beneficial.
Right, because it builds bone.
Exactly. It activates specialized cells called osteoblasts to rapidly build dense, highly fracture-resistant bones. You can run faster without shin splints, survive physical trauma, outcompete rivals for mates.
It provides an overwhelming early life advantage. You survive, you reproduce, and you successfully pass that aggressive calcium-depositing gene to your children.
Fast forward 50 years. You are now 65. That exact same gene is still eagerly promoting calcium deposition. But now it's not just depositing it into your bones. It's depositing calcium plaques into your soft tissues.
Right.
It's calcifying the smooth muscle cells in your arteries, making them stiff and brittle. It's calcifying your heart valves, severely restricting blood flow.
And the tragic catch is that natural selection is completely blind to this late-stage penalty. Natural selection cannot weed the heart disease-causing gene out of the human population because The gene already successfully did its job.
Yeah, the heart attack kills you at age 65, but the gene secured its own survival when you reproduced at age 25. Evolution doesn't care what happens to the taxicab after the passenger has already been dropped off.
It cements the reality that aging is the accumulated cost of the genes that favored our youth, combined with the lack of evolutionary pressure to repair the damage.
Well, I want to push back here on behalf of the listener because there seems to be a logical gap.
Okay, go for it.
I completely understand that evolution optimized us for a dangerous environment where you had to breed quickly before a predator killed you. But humans don't live in that environment anymore.
Fair point.
For thousands of years, we've lived in safe houses. We developed agriculture to eliminate starvation. We have sanitation. We aren't getting eaten at age 25. So why hasn't human biology caught up? If the environment is completely safe, why hasn't evolution retuned our metabolic budgets to invest in better repair mechanisms so we can live to be 200?
It's a totally intuitive question. If the selective pressures of the environment change, the organism's biology should adapt, right?
Right.
The disconnect lies in our failure to comprehend the sheer staggering scale of evolutionary time. Thousands of years of agriculture and stable societies feel like an eternity to human civilization. But on the timescale of human genetic evolution, 10,000 years is barely a rounding error.
So we haven't had enough time.
Not even close. Significant species-wide shifts in metabolic resource allocation require hundreds of thousands, if not millions of years of intense, consistent environmental pressure. The rapid development of modern medicine, antibiotics, and stable food grids has exponentially outpaced our ancient genetic budgets.
So we're essentially driving a biological machine built for a fast, dangerous sprint. But we are forcing the engine to run a cross-country marathon.
Precisely. And we can see exactly how malleable these evolutionary strategies are by looking at the extreme variations in lifespan across the animal kingdom. Ramakrishnan points out that lifespan is not a fixed physical law of the universe. It is a highly fluid evolutionary strategy.
Yeah, we see a general correlation in nature, right? Small animals with high metabolic rates live fast and die young, while larger animals with slower metabolisms live longer.
Exactly.
Take the adult mayfly. Its entire biological strategy is based on explosive reproduction. An adult mayfly doesn't even have a functioning mouth or digestive tract. Yeah, its singular goal is to hatch, frantically find a mate, reproduce within 24 hours, and die. It invests absolutely zero energy into somatic maintenance.
Contrast that with a human. We have a moderate metabolic rate, but we invest massive amounts of energy into brain development and complex social structures to protect our young. This allows us to invest more in internal maintenance, granting us a potential lifespan of 80 to 100 years.
Right.
But the outliers, the creatures that break the correlation entirely, that's where the true insights hide.
Like the Greenland shark. That's a perfect example. It lives in the freezing depths of the Arctic and North Atlantic Oceans. And the cold environment drastically lowers its metabolic rate.
It does.
It moves incredibly slowly, conserving massive amounts of energy, which it redirects into tissue maintenance. And because of this extreme evolutionary strategy, a Greenland shark can live for over 400 years. I mean, it doesn't even reach sexual maturity until it's around 150 years old.
Which is mind-boggling. But, you don't have to be a massive freezing shark to cheat the correlation. Ramakrishnan points to the naked mole rat.
Oh, the naked mole rat.
Yeah, it's a subterranean rodent roughly the size of a standard laboratory mouse. Based on its size and metabolic rate, a naked mole rat should live for 2 or 3 years.
Okay.
Instead, they routinely live for over 30 years in captivity.
Wait, how does a creature that small live that long?
Because it evolved in highly protected, deep underground colonies with very little threat from external predators. So evolution allowed it to shift its metabolic budget toward extraordinary somatic maintenance.
What kind of maintenance?
Well, their tissues produce a unique, incredibly dense form of a sugar molecule called hyaluronin. This dense sugar matrix essentially surrounds their cells and acts as a powerful anti-cancer mechanism.
Oh, interesting.
Yeah, if a cell starts to mutate and multiply, the dense matrix creates early contact inhibition, literally physically stopping the tumor from growing. They evolved a biological force field because their environment allowed them the time to do it.
And then there is the holy grail of gerontology, the hydra. A tiny freshwater invertebrate that appears to possess what biologists call negligible senescence. For all practical purposes, it does not age.
The hydra is a marvel. Its entire body is essentially composed of active stem cells that are constantly dividing and completely turning over its bodily tissues every few weeks.
That's crazy.
It gets crazier. If you cut a hydra in half, you don't get a dead hydra. The two halves regenerate into two complete, distinct organisms. It maintains perfect, continuous biological coherence.
So these outliers prove Ramakrishnan's core premise.
Yeah.
There's no universal chemical law that dictates a biological machine must rust and die at a specific time. Lifespan is merely a tunable dial on an evolutionary soundboard.
Right?
But if we accept that aging is an accumulation of damage due to evolutionary neglect, we have to transition from the theoretical why to the mechanical how. We have to dive down into the cellular level to see exactly what is breaking.
And to make the staggering microscopic complexity of a human cell understandable, Ramakrishnan employs a brilliant, overarching metaphor. He asks us to discard the image of a cell as a static, water-filled balloon, which is how it's often drawn in high school textbooks.
Right, the classic blob.
Yeah, forget the blob. Instead, imagine a cell as a vast, bustling metropolis, a city with the complexity and infrastructure of London.
Okay, I want to really build out this metaphor because it makes the invisible world completely tangible. Imagine a microscopic city containing millions of molecular citizens, all interacting with ferocious speed every single millisecond just to maintain order.
Okay, I'm picturing it.
In this cellular city, your DNA, located in the nucleus, acts as the central government. It's the highly guarded archive containing the master blueprints and laws for how every structure in the city is supposed to be built and maintained. That's good. And if the DNA is the government blueprint, the ribosomes are the massive industrial factories. They receive transcribed copies of the blueprints from the government and physically manufacture the infrastructure. They stitch together amino acids to build the proteins that do the actual physical work of keeping the city running.
Then you have the mitochondria. These are the sprawling power stations located throughout the city districts. They generate the enormous amounts of chemical energy, the ATP, required to keep the factory lights on and the machinery moving.
Got it. Power stations.
You have legions of specific repair enzymes acting as the city's maintenance crews, constantly rushing around, fixing broken pipes and patching potholes in the DNA. And finally, you have the proteasome and autophagy pathways serving as the waste management department, tasked with collecting, shredding, and recycling the metabolic garbage. It is a system of breathtaking coordination. But Ramakrishnan outlines several specific hallmarks of decay. And using the city metaphor, we can track exactly how the metropolis collapses over decades. And crucially, these hallmarks don't fail in isolation. A failure in one district triggers a catastrophic cascade in another.
Let's start at the very center, the government archives. The first hallmark of decay is genomic instability. We have this cultural perception that our DNA is a pristine, untouchable vault of information.
Oh, definitely.
But the reality is that the vault is under constant siege. The text cites Thomas Lindell, a pioneer in DNA repair, who estimated that the DNA in every single cell in your body suffers approximately 100,000 spontaneous damaging events every single day.
100,000 assaults per cell per day. That's a lot of damage.
That's massive.
The DNA molecule is inherently fragile. It is constantly bombarded by external radiation, like UV light from the sun, and internal chemical assaults from our own metabolism.
Like what?
Well, a common event is oxidation, where an oxygen radical physically alters a chemical base in the DNA, turning a normal letter of genetic code into nonsense. In our city metaphor, this is mutational vandalism. Rioters are constantly breaking into the central archives and scribbling over the master blueprints.
But when you're 20 years old, your city's maintenance crew's specialized proteins called DNA glycosylases are highly efficient. They patrol the DNA, recognize the vandalized letters, physically cut them out of the strand, and paste the correct letter back in. It happens millions of times a second without you knowing.
But as we enter evolutionary neglect, those repair enzymes inevitably slow down. They become less efficient. They start missing the vandalism.
And when errors accumulate in the blueprints, the government starts sending corrupted, garbled instructions out to the factories. The ribosomes read the broken instructions and manufacture the wrong proteins, or they manufacture them in the wrong amounts.
Exactly.
This genomic instability, the slow corruption of the information code, is the root cause of cellular dysfunction, and it's the primary initiator of cancer.
Right. And while the government archives are failing from within, there is a very specific mechanical type of instability occurring at the edges of the city limits. This brings us to the second hallmark, telomere attrition.
Telomeres.
Yeah. Telomeres are repetitive sequences of DNA located at the very ends of our chromosomes. The ubiquitous and highly accurate analogy is the aglet the little plastic tip at the end of a shoelace that stops the fabric from fraying.
Right, so telomeres stop the chromosome from unraveling and prevent the ends of different chromosomes from accidentally fusing together. But the system has a massive design flaw, known as the end replication problem.
Yeah, so the mechanism of copying DNA requires an enzyme called DNA polymerase. But this enzyme cannot start copying a strand from thin air. It requires a small primer to attach to, and it only moves in one specific direction.
Okay.
Because of this mechanical limitation, every time the cell divides and the chromosome is copied, the polymerase physically cannot replicate the very last tiny segment of the telomere.
It's like a photocopier, where the mechanical rollers always obscure the last quarter inch of the page. Every time you make a copy of a copy, the page gets slightly shorter. With every cell division, your cellular shoelace tips get ground down.
And when that protective tip is completely eroded, the bare, unprotected end of the DNA strand is exposed.
And the cell notices that, right?
Oh, absolutely. The cell doesn't just ignore this. It has a highly sensitive internal emergency alarm system. When it detects a critically short, frayed telomere, a specific protein, most notably a transcription factor called P53, is activated.
P53.
Think of P53 as the emergency brake on a train. It forcefully halts the cell cycle and pushes the cell into a state of permanent arrest known as cellular senescence. The cell goes to sleep. It can never divide again.
But from a purely defensive standpoint, the P53 alarm is brilliant. It's a vital anti-cancer mechanism. If a cell with a frayed, highly unstable chromosome continues to divide, those mutations will rapidly compound into a malignant tumor. The emergency brake saves your life in the short term.
It saves your life today, but it ensures your decay tomorrow.
Right. Because you rely on constant, healthy cell division to regenerate your tissues. You need division to heal physical cuts, to replace the lining of your gut, to maintain the elasticity of your skin. If too many cells in a tissue hit their telomere limit and are forced into permanent sleep, that tissue completely loses its regenerative capacity. The failure to rebuild is a core physical manifestation of aging. Okay, so the blueprints are corrupted and the cells are losing their ability to replicate. Let's look at the factories themselves. This brings us to the third hallmark, which is the loss of proteostasis.
Okay.
Proteostasis is a combination of the words protein and homeostasis. It refers to the cell's ability to maintain the quality and specific shape of its protein workforce.
And proteins are not just passive building blocks. They are the physical machines executing the code of life. They digest your food, they contract your muscle fibers, they serve as the receptors on your neurons.
They do everything.
Basically, yeah. But a protein is essentially a long string of amino acids. For that string to function as a machine, it must fold itself into an incredibly precise, three-dimensional shape.
And the cell has specialized proteins called chaperones, whose entire job is to surround new proteins and help them fold into the correct shape.
Right.
If a protein misfolds, exposing sticky hydrophobic parts that should be hidden inside, it becomes useless. Or worse, it becomes dangerous.
Yeah, and when a protein is hopelessly misfolded, the cell relies on its waste management system. It tags the defective protein with a tiny molecule called ubiquitin.
Like a barcode.
Exactly like a barcode. This tag tells the cellular garbage disposal, a massive barrel-shaped complex called the proteasome, to suck the protein in, shred it into individual amino acids, and recycle the parts.
But again, as evolutionary neglect sets in, the chaperones get overwhelmed, and the proteasome garbage disposals start to break down or get clogged. The waste management workers basically go on strike.
And when they strike, those sticky, misfolded proteins are left to float in the cellular environment. Because their hydrophobic parts are exposed, they try to avoid water by violently clumping together. Yeah, they form massive, insoluble aggregates. They literally form toxic clumps of molecular garbage that pile up in the streets of the cellular city, blocking the transport of nutrients and choking the machinery.
And this hallmark isn't abstract. The buildup of misfolded protein garbage is the precise physical mechanism behind some of the most devastating age-related diseases we know. Alzheimer's disease is driven by the extracellular accumulation of misfolded amyloid beta proteins and intracellular tangles of misfolded tau proteins.
Yeah.
Parkinson's disease is driven by clumps of a misfolded protein called alpha-synuclein.
It is the city slowly suffocating under the weight of its own uncollected refuse.
And clearing that refuse requires energy. Which brings us to the districts generating that energy, the mitochondria. The fourth hallmark is mitochondrial dysfunction. Your mitochondria act as microscopic power plants, generating ATP through a process called cellular respiration.
The mechanism of respiration occurs along the inner membrane of the mitochondria. through a series of protein complexes called the electron transport chain.
Sounds complicated.
Well, it functions like a hydroelectric dam. It uses the energy from the food you eat to pump protons across a membrane, creating a massive pressure gradient. When those protons flow back through a molecular turbine called ATP synthase, it generates the chemical energy that powers every single action in your body.
But Ramakrishnan explains that this turbine system is inherently dangerous.
Yeah, it's like running a heavily polluting coal power plant inside a densely populated neighborhood. The electron transport chain is not perfectly sealed. As electrons are passed down the chain, some of them slip out and inadvertently interact with the oxygen you are breathing.
Okay.
This creates a highly volatile byproduct called reactive oxygen species, or ROS. These are free radicals.
They act like microscopic sparks flying out of the furnace. And because the mitochondria have their own separate circular DNA located right next to the furnace, those sparks constantly bombard and mutate the mitochondrial blueprints.
This creates a vicious cycle. The sparks damage the mitochondrial DNA. The damaged DNA produces faulty turbine proteins. The faulty turbines become less efficient at making energy and leak even more sparks.
Oh, wow.
This is the foundation of the oxidative stress theory of aging. Over time, the mitochondria swell, their energy output crashes, and the cellular city experiences massive rolling blackouts.
I want the listener to physically internalize this because it is the ultimate biological irony. Think about the physical act of taking a deep breath right now. The oxygen entering your lungs, traveling through your bloodstream, and entering your mitochondria is the exact fuel keeping your consciousness online.
Yes.
But the chemical reality of processing that oxygen is simultaneously producing the oxidative sparks that are slowly rusting your cellular engines from the inside out. The absolute necessity of breathing is the very mechanism driving your degradation.
It's an inescapable thermodynamic catch-22. And as those dying mitochondria leak stress signals, they tie directly into the final and perhaps most destructive hallmark, cellular senescence.
We mentioned earlier that when a cell's telomeres wear down, or when it sustains too much DNA damage, the p53 brake is pulled and the cell goes into permanent sleep.
Right.
But Ramakrishnan details how this sleep is a massive misnomer. These senescent cells don't just sit there quietly. They transform into what gerontologists literally call zombie cells.
Yeah. They refuse to undergo apoptosis and die gracefully. They linger in the tissue matrix. And as they sit there, their internal programming goes completely haywire.
What do they do?
They begin aggressively secreting a highly toxic cocktail of inflammatory cytokines, chemokines, and tissue-destroying proteases. This massive secretion is known as the senescence-associated secretory phenotype, or SASP.
So if we return to our city metaphor, these aren't just empty, sleeping buildings. They are decaying, abandoned chemical plants that have started actively leaking toxic radioactive sludge into the surrounding neighborhood.
Exactly. And that SSP sludge fundamentally alters the local environment. It poisons the neighboring, perfectly healthy cells, triggering them to undergo premature senescence as well.
So it's infectious.
The zombie infection literally spreads outward from the initial damaged cell. This creates a state of chronic, systemic, low-grade inflammation throughout the entire body. It is a phenomenon so pervasive that researchers coined the term inflammaging.
Inflammaging.
It degrades the extracellular matrix, destroys cartilage, and creates an environment highly conducive to tumor growth.
So to bring Ramakrishnan's incredible metaphor to its conclusion, aging is not a sudden, pre-programmed stroke of midnight. It's a slow, agonizing loss of systemic coherence. The government archives are vandalized by mutation. The shoelace caps burn away, halting the ability to rebuild. The ribosomal factories choke on misfolded protein garbage. The mitochondrial power grids rust and trigger rolling blackouts. And the zombie buildings flood the streets with inflammatory sludge.
That's the bleak reality.
Eventually, the communication networks fail entirely. The city collapses. He beautifully and tragically compares it to the ancient, ruined city of Hampi in India. It's a structure of undeniable past glory that simply could not maintain itself against the erosion of time.
It really forces us to confront our fragility on a mechanical level, but it also illuminates exactly why intervening in this process is so monumentally difficult.
And that difficulty is the perfect pivot point for our deep dive.
Yeah.
Because while biologists look at that crumbling cellular city and marvel at the complexity, there's an entire, heavily funded industry of people who look at the ruins of Hampi and think, I could just write a software patch to fix that.
Yeah, the tech bros.
I'm talking about the modern longevity industry. Let's look at the hubris of trying to hack human biology.
Ramakrishnan is incredibly sharp and frankly quite critical when analyzing this sector. He describes the current state of the longevity industry as fevered. Tens of billions of dollars are being funneled into biotech startups, primarily driven by aging tech billionaires from Silicon Valley. And Ramakrishnan diagnoses a fundamental philosophical flaw in their approach, which he identifies as the software fallacy.
The software fallacy. Let's get into the mindset of a tech entrepreneur. If you build your empire in software, you're accustomed to a world of logical top-down design. If an application crashes, you know there's a specific error in the code. You locate the bug, you write a clean patch, you deploy it, and the app runs perfectly again.
Right.
Because they view the world through this lens, they view biological aging merely as a technical problem. It's just buggy legacy code that needs to be debugged.
But Ramakrishnan forcefully reminds the reader that biology is emphatically not software. Code is created intelligently. Biology is a chaotic, bottom-up kludge. It is. It's the result of billions of years of random genetic mutation combined with whatever duct tape solutions helped an organism survive slightly longer in the Pleistocene era. Biology is incredibly redundant, meaning multiple pathways control the same outcome. It is highly stochastic, meaning molecular interactions rely heavily on random chance and probability.
And as we learned with antagonistic pleiotropy, it's built entirely on evolutionary trade-offs.
Exactly. You cannot isolate a variable in biology the way you can in a computer program. If you pull one metabolic lever to supposedly fix a problem in the mitochondria, you will inevitably trigger 3 unforeseen, catastrophic cascading effects in the immune system. You cannot cleanly debug a system that possesses no underlying rational logic.
Ramakrishnan also raises a massive ethical and structural concern regarding the culture of this industry, which is severe financial conflicts of interest.
This is where Ramakrishnan's position as an independent academic Nobel laureate is crucial. He notes a disturbing trend. Many of the world's most prominent gerontologists, the scientists actually running the labs and publishing the foundational papers on aging, now hold massive financial stakes in the biotech startups they're advising.
Oh, wow.
Yeah, they're sitting on corporate boards. They are receiving massive equity packages.
Right, and when the scientist validating the drug is also the shareholder who becomes a billionaire if the drug goes to market, the scientific method gets compromised by the hype machine.
It is inevitable. When billions of dollars in venture capital are on the line, the public relations announcements and the promises of curing death radically outpace the actual rigorous, peer-reviewed clinical data. Ramakrishnan observes the irony of these individuals. They spent their entire youth obsessively working to become unimaginably wealthy, and now they are desperately trying to spend that wealth to buy back their youth.
But biology doesn't care about your stock options.
It really doesn't. Biology does not respond to venture capital.
So let's examine what that venture capital is actually producing. The source material evaluates several specific biological interventions that are currently the darlings of the longevity landscape.
Right.
Let's run through the mechanisms of what they're actually trying to do. The first major contender is a drug called rapamycin.
From a purely mechanistic standpoint, rapamycin is fascinating. It was originally discovered in soil samples from Easter Island. In the cell, rapamycin targets a highly conserved protein complex known as the MTO pathway. Yeah, mTOR acts as a central nutrient sensor for the cell. When nutrients and amino acids are abundant, mTOR signals the cell to grow, build proteins, and divide.
But rapamycin acts as an inhibitor. It physically binds to mTOR and shuts it down.
And when mTOR is inhibited, the cell is tricked into thinking it is starving, even if nutrients are present. This perceived starvation forces the cell out of a growth phase and triggers a defensive maintenance phase.
Oh, right.
Crucially, it massively upregulates autophagy, the waste management system we discussed earlier. The cell starts aggressively hunting down misfolded proteins and damaged mitochondria, consuming them for spare parts. Rapamycin essentially mimics the longevity benefits of severe caloric restriction without the organism actually having to starve.
And in the lab, this works spectacularly, right?
In animal models, yes. It is one of the very few compounds that has consistently been shown to extend both the median and maximum lifespan in mice, worms, and flies. However, the translation to human longevity hits a massive biological wall. Rapamycin is primarily used in human medicine as a potent immunosuppressant. It is given to organ transplant patients precisely because it prevents T cells from proliferating, stopping the body from rejecting the new organ.
Oh, so the trade-off is brutal. A healthy 50-year-old might start taking rapamycin to trigger autophagy and clean out their cellular garbage, hoping to live to 120. But in doing so, they deliberately suppress their immune system, making them incredibly vulnerable to bacterial pneumonia or a viral infection. You might successfully slow down the biological clock, only to die from a common cold.
It perfectly illustrates the danger of pulling a single lever in a complex system. Let's look at another highly publicized drug, metformin.
Metformin is entirely different. It's an incredibly cheap, generic drug that's been prescribed for decades to treat type 2 diabetes by lowering blood glucose levels. Why is the longevity industry obsessed with the diabetes drug?
Researchers noticed a strange statistical anomaly in massive epidemiological databases. Diabetic patients taking metformin seem to be living slightly longer and experiencing lower rates of certain cancers and cardiovascular events than non-diabetic individuals who weren't taking the drug.
That's weird.
Right. Mechanistically, metformin mildly inhibits complex I in the mitochondria. This slight energy stress activates an enzyme called AMPK, which, similar to rapamycin, shifts the cell's metabolism toward maintenance and reduces systemic inflammation. There are currently massive clinical trials underway to see if giving metformin to healthy adults actually delays the onset of age-related diseases.
Let's move from metabolic sensors to something much more aggressive. There's a new class of drugs called senolytics. The name is a combination of senescence and alytic, which means to destroy.
Senolytics are precisely targeted weapons. They are designed to hunt down and induce apoptosis in those zombie cells. We discussed the senescent cells that refuse to die and continually pump out toxic inflammatory SSP sludge. Drugs like dasatinib, which is a leukemia medication, paired with quercetin, a plant pigment, have been shown to selectively clear these zombie cells from the tissues of mice, resulting in remarkable rejuvenation of physical function.
On paper, this sounds like an absolute victory. Send in a drug, assassinate the zombies, stop the inflammaging, and clean up the neighborhood. What is the biological catch?
The catch is that biology rarely creates a mechanism that is purely evil. Senescence is not just a mistake. It's a vital physiological state used for specific tasks, particularly tissue repair and wound healing.
Okay, wait, explain that.
Well, when you suffer a severe physical injury, specific cells temporarily enter senescence and secrete factors that actively summon immune cells to the injury site to begin the rebuilding process.
Oh, so if a billionaire takes a heavy dose of senolytics to clear out all the aging cells in their body, they might significantly reduce their systemic inflammation. But if they subsequently get into a car accident or suffer a severe laceration, their tissues might completely fail to initiate the wound healing process. They trade long-term tissue maintenance for acute vulnerability.
Exactly. The final intervention evaluated in the text is undoubtedly the most sensational and, frankly, the most macabre. It is the pursuit of young blood. The clinical scientific term is parabiosis.
The headlines around this are pure science fiction. Billionaires harvesting the blood of the youth. Where does this actually originate scientifically?
It originates from deeply unsettling laboratory experiments where scientists physically surgically join the circulatory systems of a young mouse and an elderly mouse. They literally stitched their flanks together so that the heart of the young mouse pumped blood through the organs of the old mouse and vice versa.
I mean, that's wild. And the results were startling enough to launch a dozen startups.
They were. The researchers observed significant signs of physiological rejuvenation in the old mouse. Its muscle tissue repaired faster, its neural stem cells showed increased activity, and its fur grew back thicker. The immediate hypothesis was that the plasma of the young mouse contained specific, unidentified youth factors, hormones or proteins, that actively reverse the aging process in the old tissues.
Because the Silicon Valley mindset is move fast and commercialize, this single observation in rodents immediately led to the creation of what the media dubbed vampire clinics. Wealthy individuals began paying thousands of dollars for intravenous transfusions of plasma harvested from young, college-age donors, hoping to mainline those mysterious youth factors.
But Ramakrishnan remains intensely, rigorously skeptical of this entire enterprise. First, the human clinical trials attempting to replicate this with plasma transfusions have failed to demonstrate any miraculous sustained reversal of aging. But more importantly, there is a massive confounding variable in the original mouse experiments that the startups conveniently ignore.
The dilution effect.
Precisely. When you join the circulatory systems, you aren't just giving the old mouse young blood. You are significantly diluting the old mouse's own toxic inflammatory blood.
Oh.
Yeah, recent studies suggest that the rejuvenation isn't caused by magical youth proteins in the young plasma. It's simply the result of filtering out the accumulated SASP factors and metabolic garbage that was suffocating the old mouse's organs.
So you could just clean the blood.
You might achieve the exact same result through therapeutic plasma exchange, essentially just filtering the patient's own blood without needing a young donor at all. It is a textbook example of commercial hype completely ignoring the nuances of the scientific method.
Listening to this litany of failed trade-offs. If I'm a hyper-successful tech founder who is used to brute forcing my way through obstacles, I'm sitting here thinking, fine, biology is messy, senolytics ruin wound healing, rapamycin ruins the immune system, but surely this is just a computational problem. If I fund massive AI supercomputers to perfectly model every protein interaction and I throw $50 billion at CRISPR gene editing, shouldn't we eventually find a way to safely clear the zombies, perfectly rewrite the DNA, and just engineer our way out of this?
It is the ultimate expression of techno-optimism, right? The belief that with enough processing power, biology will submit. But Ramakrishnan answers that hypothetical by revealing the ultimate terrifying biological wall that blocks the path to immortality. It is the deep, fundamental, inescapable biological link between aging and cancer.
This is the ultimate catch-22 of human existence. The very things that kill us slowly are the exact things preventing us from dying quickly.
The fundamental trade-off of multicellular life is this. The precise cellular mechanisms that we desperately want to turn off or bypass in order to stop the aging process are the exact same mechanisms that act as our primary defense against malignant tumors.
Let's look at the absolute cutting edge of longevity research to see how this terrifying paradox plays out. The holy grail right now is cellular reprogramming. This field was born from the revolutionary work of another Nobel laureate, Shinya Yamanaka.
Yamanaka made a discovery that fundamentally rewrote our understanding of cellular destiny. He identified 4 specific transcription factors, proteins that bind to DNA and control which genes are turned on or off. By introducing just these 4 factors, now known as the Yamanaka factors, he proved you could take a fully mature, specialized adult cell, like a skin cell, and completely reprogram it backwards.
We need to explain how profound this is. As a cell matures, its DNA is marked with chemical tags, primarily methyl groups, in a process called epigenetics. Think of the DNA as a massive million-page textbook. A skin cell has thousands of pages permanently glued shut by epigenetic tags, so it only reads the chapter on being skin. It can never be a liver cell. Yamanaka found a way to dissolve the glue.
He found the chemical reset button. Applying the Yamanaka factors aggressively strips away those epigenetic methylation marks. It wipes the cell's memory, reverting it back into a pluripotent stem cell.
A blank slate.
Exactly, a completely blank slate, identical to the cells found in an early-stage embryo, capable of dividing indefinitely and becoming any tissue in the body.
The theoretical application for longevity is staggering. You could theoretically bathe an aging, failing human liver in a precisely calibrated dose of these factors, wipe away the epigenetic damage of time, and revert the organ back to a biologically pristine 20-year-old state. You could literally reset the clock.
That is the multi-billion-dollar dream. But Ramakrishnan forces us to look at the nightmare reality of what happens when scientists actually attempt this in living mammalian tissues.
What happens?
When you forcibly strip away a cell's identity and turn it back into a pluripotent stem cell, you are handing that cell the power to divide rapidly and uncontrollably. You are removing its specialized restraints. In countless laboratory experiments, when reprogramming is initiated in vivo, the cells don't neatly and politely regenerate a fresh organ.
So we guess they go crazy.
They panic. They multiply wildly and form teratomas.
Okay, we have to describe what a teratoma is because it highlights the sheer chaos of biology that software engineers fail to respect. It is biological horror.
A teratoma is a chaotic, monstrous tumor driven by out-of-control, pluripotent stem cells. Because the cells have no epigenetic instructions telling them what to be, they start randomly differentiating into whatever tissue they want, wherever they are. You attempt to rejuvenate a mouse's kidney, and instead, you trigger a rapidly expanding tumor inside the kidney that contains random patches of fur, fully formed teeth, beating cardiac muscle cells, and shards of bone.
Yeah, that's horrifying. Reverting a cell to its youth is an incredibly delicate high-wire act. And what the data shows is that attempting to walk that wire, almost inevitably, violently tips over into malignancy.
We see the exact same terrifying paradox when we look at telomeres. Remember the cellular shoelaces we discussed?
Right. Every time the cell divides, the telomere gets shorter until the p53 alarm is triggered and the cell becomes a senescent zombie. Naturally, everyone in the longevity space wants to design a drug that universally activates telomerase.
Which is the enzyme that regrows them.
Exactly. Telomerase is an enzyme, specifically a reverse transcriptase, that carries its own RNA template and can physically stitch new DNA back onto the ends of the chromosome. The logic is incredibly seductive. If we activate telomerase, we constantly regrow the protective caps. The cells never hit the alarm, they never turn into zombies, and our tissues retain the ability to regenerate forever.
The logic is seductive, but the biological reality is lethal. Ramakrishnan points out a dark, undeniable truth about oncology. Universally activating telomerase is precisely what cancer cells do to become immortal.
Wait, really?
Yes, a microscopic mutated tumor is naturally limited by its telomeres. It will eventually divide itself into senescence and stop growing. A tumor only becomes a lethal systemic threat when it undergoes a secondary mutation that reactivates the telomerase gene. This allows the cancer cells to endlessly rebuild their telomeres, completely bypass the p53 emergency brake, and divide infinitely until they crush the host's organs.
So if you take a pill that artificially elongates the telomeres in every cell in your body, you are systematically dismantling your body's primary evolutionary defense mechanism against tumors. You're actively handing any dormant microscopic precancer cell in your tissue the exact biological tool it needs to become an unstoppable malignancy.
It's the ultimate backfire.
I want to synthesize this terrifying reality for the listener with an analogy. Trying to cure human aging by universally activating telomerase is essentially like looking at your car and deciding, I want to go 300 miles an hour forever. So you install a massive rocket-powered turbo engine. But to make room for the engine, you completely rip out the brake lines and the steering wheel. Yes, when you hit the gas, you're going to go incredibly fast. You will feel phenomenally powerful, right up until the exact moment you slam into a brick wall at terminal velocity.
That analogy perfectly captures the mechanism. You remove the brakes of senescence and you crash into the wall of cancer. The biological corridor between decaying into old age and exploding into malignant tumors is agonizingly narrow. True immortality requires a level of simultaneous perfect control over trillions of independent stochastic cellular events that may simply be mathematically impossible.
Well, we've covered the sheer mechanics of the cellular metropolis, the evolutionary economics of the soma, and the terrifying medical brick walls of cancer. But as we enter the final act of this deep dive, Ramakrishnan requires us to pan the camera back.
Right.
We have to transition from the biological barriers of immortality to the profound philosophical and ethical barriers.
Yeah, the big questions.
Because suppose, purely for the sake of a thought experiment, the tech billionaires actually win. Suppose an artificial intelligence models protein folding so perfectly that we invent a gene therapy that clears the zombies, regrows the telomeres, and resets the epigenetics without triggering a single teratoma or cancer cell. We cure death. What actually happens to human civilization?
Ramakrishnan tackles this directly. Even if we possess the theoretical capacity to extend human life to 200, 500, or 1,000 years, should we execute it? What is the unseen societal cost of biological stagnation? The first major threat he identifies is the end of generational turnover.
Death serves a brutal, emotionally agonizing function, but on a societal level, it's a necessary engine for progress.
Exactly. If you look at the history of science, civil rights, or political philosophy, rapid innovation rarely occurs because the entrenched, older generation suddenly experiences a massive paradigm shift and changes their minds. Progress occurs because the older generation physically passes away, and a new generation who are not psychologically wedded to the old dogmas and power structures takes their place.
Makes sense.
Max Planck, the legendary quantum physicist, famously and morbidly observed that a new scientific truth does not triumph by convincing its opponents. It triumphs because its opponents eventually die. Science advances one funeral at a time.
Imagine a world ruled by a permanent class of immortal tech billionaires, immortal politicians, and immortal corporate board members. They would accumulate compounding wealth and dictate policy for centuries. They would never relinquish power. You'd completely suffocate the dynamism, the turnover, and the fresh perspectives that allow human society to evolve. Our culture, our science, and our political structures would become permanently trapped in amber.
And tying directly into the consolidation of power is the terrifying economic prospect of the immortality gap. We have to be brutally realistic about how medical technology is distributed. If a genuine, safe, comprehensive life extension protocol is invented, it's not going to be a cheap, generic pill you pick up at the local pharmacy.
Definitely not. It'll be a highly bespoke suite of continuous interventions. Regular stem cell infusions, constant epigenetic monitoring, CRISPR gene therapies to correct accumulating mutations.
It will cost millions of dollars a year to maintain, which means the cure for aging will create a literal biological bifurcation of the human species. You will have a biological elite, the ultra-wealthy, who can afford to purchase centuries of pristine, disease-free life. And you will have a vast, disenfranchised lower class that remains bound by natural mortality, continuing to suffer from Alzheimer's, heart disease, and decaying tissues, dying at 80.
Throughout all of human history, despite the massive inequalities in wealth, power, and comfort, the one great, unavoidable equalizer between the emperor and the peasant was the grave. Both of their temporary vessels eventually failed. If you invent a technology that removes that final equalizer for a select few, you magnify societal inequity to an unprecedented and likely irreversible degree. You literally codify economic class division into the human genome. It is a chilling dystopia.
But Ramakrishnan doesn't just rest on the macro-level societal dystopias. He takes the ethics question down to a deeply personal philosophical level. He asks us to consider whether the conscious knowledge of death, that heavy psychological burden we discussed at the very beginning of the show, is actually the foundational source of life's beauty and meaning.
He shares a very poignant personal anecdote in the final chapters of the book. He describes taking a quiet walk in Hardwick Wood, an ancient, sprawling forest in the UK. And while walking among these massive, silent, enduring trees that have stood for centuries, he comes across a small, physical memorial. It's a tribute to a young man named Oliver John Hardiman, who died at the age of 25.
He uses that striking visual contrast, the fleeting, brief spark of a 25-year-old human life, set against the slow, silent, almost immortal endurance of the ancient wood to reflect on the nature of our existence. He invokes the writings of the poet Rabindranath Tagore, who observed the butterfly.
The butterfly.
Yeah, a butterfly's life is beautiful precisely because it is so incredibly fleeting. The fragility, the absolute lack of permanence, is the core source of the beauty.
It raises the ultimate question about human motivation. If you remove the ticking clock of mortality, do we fundamentally lose the drive that makes us human? Does the urgency to discover a new continent, to love fiercely, to write a symphony, to build a legacy, simply evaporate if you have endless centuries to get around to it?
That's the question.
I want you, the listener, to think about how you physically experience time. Think about how you approach your weekends. Let's say you are roughly 30 years old right now. Mathematically, you have maybe 2,500 weekends left in your entire life. You treat those weekends a certain way. You plan trips, you visit family, you try to extract joy from them because you subconsciously know the supply is strictly, mercilessly limited. But imagine if a doctor handed you a pill and suddenly gave you an infinite supply of weekends, an endless, sprawling expanse of time with absolutely no deadline. Would you ever actually get off the couch to write that novel? Would you ever take the risk of asking someone to marry you if you knew you had 4 centuries to figure it out? Does the looming presence of the deadline create the masterpiece?
It is the psychological manifestation of the economic principle of scarcity. Value is entirely derived from limits. Without the boundary of death, an infinite life might degrade into an endless, shapeless plateau of procrastination and apathy. The urgency of the temporary vessel is what generates the heat of human experience.
It brings us to the ultimate synthesis of Venki Ramakrishnan's incredible work. In Why We Die, he has taken the most profound, historically terrifying mystery of our existence and laid the mechanics completely bare. By stripping away the poetry and the theology, he shows us that death is not a preordained script. It isn't a malicious, active program designed by evolution to punish us for existing.
It is simply the inevitable thermodynamic consequence of being a breathtakingly complex but imperfect biological machine. We are temporary vessels designed by a blind, resource-constrained evolutionary process to protect and transport the torch of genetic information across the vast, hostile expanse of time, handing it off to the next generation before our own machinery inevitably rusts and fails.
And while the science of biogerontology is making incredible strides and may very well provide us with interventions that significantly extend our health span, meaning we might live those 80 or 90 years with much less chronic disease, less joint pain, and clearer minds, the techno-optimist quest for absolute biological immortality remains a distant and perhaps deeply biologically flawed dream. Understanding the cold, hard molecular basis of our mortality, Knowing about the slipping telomeres and the oxidative sparks doesn't strip life of its meaning. If anything, recognizing the absolute statistical miracle of the cellular city working at all, the fact that millions of proteins are folding perfectly inside you right now, just so you can comprehend these words, highlights the extraordinary, fragile ballet that allows us to exist in the first place.
It teaches us the profound wisdom of accepting the temporary vessel. Life is an active, burning process of furious repair, unimaginable cellular cooperation, and eventually a necessary, graceful yielding to the thermodynamic realities of the universe.
Well, thank you for joining us on this deep dive into the molecular machinery of mortality. We hope we've given you a radically new lens through which to view your own incredible biology and a deeper appreciation for the trillions of cells working tirelessly to keep your city running. But before we go, I want to leave you with one final provocative thought to mull over.
Let's hear it.
We've talked extensively about how the ticking clock of physical decay gives life its urgency. But imagine a future where the biotech billionaires partially succeed. They realize they cannot cure death because the cancer wall is too high, but they completely perfect health span.
Okay.
Imagine a world where specific, targeted medical interventions ensure that your cellular metropolis never crumbles.
Yeah.
You stay physically, biologically, and mentally feeling exactly like a vibrant 30-year-old until the midnight of your 100th birthday. No arthritis, no dementia, no frailty. And then at exactly 100 years old, your temporary vessel simply runs out of telomeres and cleanly shuts down entirely overnight. If the slow, agonizing physical decay of aging was completely removed from the human experience, but the hard, absolute deadline of death remained exactly the same, how would that change the way you plan the final decade of your life? Without the accumulating aches, pains, and fatigue to subconsciously signal to your brain that the end is approaching, would you finally be at peace with the boundary? Or would that sudden sheer drop-off stepping directly from perfect, vibrant health into the absolute void make the absolute certainty of death even more terrifying to bear?
It brings us right back to the psychological burden we started with.
It really does. We may have traded the poetry of the stars for the precision of the electron microscope. We may have mapped the genome, tracked the telomeres, and categorized the misfolded proteins. But no matter how deep into the chemistry we dive, we still end up standing at the edge of the exact same philosophical cliff. We'll leave you to look over the edge.